Hereby, I open this academic ceremony in which Benjamin Wilde will defend the academic thesis, The Kidney and the Adaptive Immune System in ANCA Vasculitis, Attack versus Regulation. My name is Harry Schouten. I'm an emeritus professor of hematology and the pro-rector today. Welcome everyone in the audience. May I invite you to present a summary of your study and the conclusion of your thesis.
Dear Pro-Rector, dear Corona, dear supervisors, dear family and friends, dear colleagues, thank you very much for joining my thesis defense today. I will now lay out the content of my thesis in some sort of layman terms. So ANCA vasculitis is a very severe disease. It is an autoimmune disease and it affects mainly the small vessels of the body, and it is characterized by the presence of antibodies. These are proteins that are directed against molecules of your own body. In this case, it is directed against myeloperoxidase and proteinase 3. And the problem with this disease is if it is left untreated, it always has a fatal disease course. And in up to 80% of the cases, the initial presentation is with glomerulonephritis. So the clinical manifestations are quite diverse because it's a disease that affects the small vessels. You find the small vessels everywhere in the body, and basically every organ can be affected, but the most common presentation is indeed with glomerulonephritis. There are some unmet medical needs, so there are some things that needs to be solved in ANCA vasculitis. We have patients that do not fully respond to the first therapy that we give to them, that's the so-called induction therapy. And even if we have patients that respond to the first initial therapy and they enter remission, we have patients that fail to keep the remission during their disease course, so they relapse even if they are further treated. A quarter of the patients die within the first 5 years of the diagnosis, so it's a fatal disease, and 50% of the death within the first year that occur after diagnosis are due to infections. So there are a lot of issues that needs to be solved in this disease, and the best way to solve disease issues is always to understand how a disease works. And it's like a puzzle basically, and the pathogenesis of ANCA vasculitis has been, in contrast to other autoimmune diseases, unraveled to a valuable extent so far. And I will tell you something about the relation of attack and regulation in this disease. So this disease is mediated also by parts of the immune system that we all have, so by the cellular immune system. And the cellular immune system has parts that attack, so that defend the body actively. But if you have attackers, you also need regulators that limit the attack so that no harm occurs. And I will tell you something about the regulators first, how the regulators regulate in this disease and how they may fail. And I will also tell you something about the attackers and how the attackers may evade a little bit of the regulation in this disease. So the first thing that I want to talk about is dendritic cells. So if you have a look at kidney biopsies in patients that have active disease, then you will find a lot of different immune cells within the kidney. And you will also find immune cells that have properties and act as orchestrators of the immune system. These are the dendritic cells, among other populations. And we find a lot of dendritic cells in the kidney of patients, and these dendritic cells show some properties that suggest that these cells interact within the kidney with T cells, so that they orchestrate and regulate T cells within the kidney, and we think that these cells are important to locally regulate the immune response in this disease. So these are regulators, and we find them in vast numbers in the kidney. We also have other systems that work within the kidney to limit and to steer the cellular immune response. And next to dendritic cells, we find a lot of T cells within the kidney, and these T cells would not be there under normal circumstances. In this disease, they are in the kidney, and mostly we think they are doing harm. And to prevent that T cells are doing harm in the kidney, there are specific systems that limit T cells. These are T cell breaks, and these T cell breaks are called co-inhibition. And we see that the co-inhibition factor PD-1 that is expressed on T cells is there in patients. But if you try to functionally assess the T cell break, so is the T cell break really working as well in patients as in healthy control, then we see that the T cell break is not as efficient in patients as in contrast to healthy controls. If you look at the T cell break PD-1, and most importantly, the T cells that we find in the kidney in patients, they do not express PD-1. So they cannot be regulated within the kidney with PD-1. So we had a look at another T cell break, BTLA, and also BTLA is expressed on circulating T cells like PD-1. But in contrast to PD-1, BTLA is fully working in patients. So if you assess the functionality of BTL1 in patients and compare it to healthy controls, then we basically see that the functionality is there. Furthermore, what we also find is that subpopulation of dendritics of T cells that are very important in this disease, the so-called Th17 cells, that also these Th17 cells are quite well regulated by BTLA in patients as well as in healthy controls. So we have two T cell breaks, two important regulators. One is working, BTLA, and one is not working, PD-1. We had a look at cells that have no other duty than being a regulator. And one of these cells are the so-called regulatory B cells. These B cells limit T cells. They can steer T cell responses. This is a little bit unexpected because in previous times it was always thought that B cells are merely antibody producers, and that's what they do, and maybe a little bit of antigen presentation. But indeed, they can produce cytokines. These are proteins that are secreted by the cells that directly suppress T cells. And we had a look at B cells that suppress via IL-10, and we find that these IL-10 producing BRACs are reduced in patients in remission as compared to healthy controls. But if you look at patients that have active disease, also in these patients, these circulating regulatory B cells are reduced in contrast to healthy controls. Furthermore, we found that not only IL-10 is an important mediator of regulatory B cell function, but that there's also a specific subset that produces granzyme B, which is basically something that can directly kill under specific circumstances. So we have cytotoxic B cells that have regulatory function, and these cytotoxic B cells exist as a separate subpopulation next to these IL-10 producing regulatory B cells. We always think that there must be a specific balance of attackers and regulators, so we also have to have a look at the attackers. So far, we just discussed the regulators. So if we look at the attackers, there's one specific subset that needs to be mentioned. These are the so-called Th17 cells. And the special idea about these Th17 cells is that they have a specific role also in kidney inflammation. Basically, the renal parenchymal cells are able to produce chemokines that are able to specifically attract Th17 cells to the kidney and then being the starting point of renal inflammation. These Th17 cells promote autoantibody formation, so antibodies that are directed against your own body. These Th17 cells promote tissue inflammation within the kidney, and they also promote destruction of the kidney. And in summary, they have a detrimental impact on the kidney. So we had a look at these Th17 cells in patients with ANCA vasculitis, and we assayed these cells in patients that were lacking disease activity, so patients in remission, and compared them to healthy controls, and we saw that these circulating Th17 cells were highly elevated in patients in remission. And if you look at patients that have signs of active disease, so that were suffering from active ANCA vasculitis, then we also saw an expansion of these Th17 cells. So the numerical increase of these cells that we see is independent of disease activity, and it persists. So the question was now, can we target these Th17 cells? Can we target these attackers to basically make disease better? And for this issue, we applied an animal model of ANCA vasculitis. And in this case, we made use of Vista Kyoto rats, and we induced disease in these rats with a specific scheme of injections. And after 6 weeks, we culled the rats to see what the disease was doing in specific organs. And here you see the features of the model that we have. So we see that basically all of the animals develop anti-MPO antibodies. We see that they have mild kidney disease, so that is depicted here. That's the number of abnormal glomeruli that we find in each animal. And then as a readout for renal damage, we always look at the protein secretion in the urine. That's the albuminuria, and the albuminuria is at the endpoint of the study elevated as compared to the control animals. And we see in this model even pulmonary vasculitis, which is a specific feature also of this model. So it's not only limited, restricted to the kidney, we also see vasculitis in other organs. And now we looked at Th17 cells, and we saw that over the course of the disease, Th17 cells enter the kidney of these rats. That can be appreciated here. So the numbers of renal Th17 cells is highest at the endpoint at week 6. And interestingly, these Th17 cells are also specific for the antigen these animals were immunized against, MPOs. So these Th17 cells had a target that they followed into the kidney, MPO. So our question was then, can we basically treat these animals with just neutralizing the key cytokine IL-17A? And for this purpose, animals were treated beginning before disease induction. That was one strain, and the other group was treated after disease was already established, and then for 3 weeks with regular injections of anti-IL-17 antibody. So, and what we basically saw is that the treatment of these animals was not efficacious, so it didn't work. So it did not prevent the development of anti-MPO antibodies, and it did not prevent the onset of kidney disease, and it did not prevent the onset of pulmonary vasculitis. And based on these results, we just concluded that the neutralization of one single cytokine is just not a good idea in this disease. And with this, I already want to summary and conclude. So we had a look at regulation in ANCA vasculitis. We saw that we have regulation within the kidney itself that is steering the immune response. But we also see that specific regulatory pathways are defective. They are simply not working in ANCA vasculitis. But we also have functional regulatory pathways, and maybe we can use them as a therapeutic target. We had a look at the attackers in ANCA vasculitis, and we saw that circulating pro-inflammatory Th17 cells were expanded. We saw that these T cells also enter the kidney and destruct the kidney, but it's not done with just eliminating the key cytokine IL-17A. It must be probably a more combined therapeutic approach. With this, I want to end and give back to the pro-rector.
Thank you for your presentation. The opposition will be opened by Professor Schurgers, Professor of Biochemistry of Vascular Calcification at this university and Chair of the Assessment Committee.
Thank you, Mr. Pro-Rector. Dear candidate, with great pleasure I have read your thesis. It's not completely my topic, so I had to really dive into it. Thank you for that. It's a tremendous amount of work. You have 10 chapters, 10 publications, and that these 10 is only 10 out of 100 plus publications. And then one might wonder what happens if you have so many publications, but it took you from 2009, your first publication, to 2026, why you waited. You have a very heavy supervision team. I don't know if that is because you're a difficult person or the tremendous amount of work, but I think if we read your work, then we have a true scientific diamond here. And with these congratulations, of course, I want to also extend that to the supervision team. Let's start with chapter 1, because as I said, you started in 2009. We are now 2026, and also in your chapter 1, which is from 2011, you updated the chapter. It's the introduction on the background and the disease mechanisms and the treatment of ANCA vasculitis. Can you briefly tell me what has changed over 16 years of research?
Yeah. So as I thank you, highly esteemed opponent, for this question and also for your congratulations. Thank you also for this question. A lot has changed, and that's due to the fact that the puzzle of the pathogenesis has been completed by many, many other researchers and many, many groups. And basically we moved on from unspecific targets, from targeting the disease with cyclophosphamide, which is basically a very broad immunosuppressive drug, to treating specific targets like B cells. So rituximab, I think, was approved, at least in Germany, for treatment in 2013. So I mean, only 14 years ago, but that tells us already that it took a while till the pharmaceutical companies also gained interest. So rituximab was the first drug that was then approved for specific targeting. And after that, we only had one additional drug that was approved for therapy, and that is avacopan, basically. And
that is a little bit disappointing if I hear that.
It is.
Yes. And so how many years do we still need then before we cure ANCA vasculitis?
Well, I think we are now in lucky times, because the pharmaceutical companies and also the little pharmaceutical startups, they really gain interest in this disease because they see that there are a lot of translational approaches done in that field that can be translated successfully into therapies like with avacopan. And I think avacopan was a prime example for that, because the group of Peter Heringer basically developed the concept of complement blockade in this disease, and it worked quite well. Yeah. Basically, it proved really well in clinical trials, and if you now look at the ongoing clinical trials in ANCA vasculitis, so if you look at clinicaltrials.gov, there are a lot of phase 2 trials going on now, but you also have to keep in mind that it's a rare disease. It's not like rheumatoid arthritis or psoriasis arthritis, so it's not that easy to recruit patients, and it is also not wise to have a lot of competing trials.
Yeah.
So
I believe you, because time is limited, and I'm very sort of pro-rector who will stop me. So you choose the kidney, and I love that, because that's the connection to my work a little bit, because kidney disease is also having a vascular disease. Why the kidney? Why not the lungs or the skin? So why was it ANCA vasculitis and kidney? Just very brief answer.
Yeah, so basically I started science in nephrology when I was in Essen, and that was why I got in touch with ANCA vasculitis at first. So the kidney was always at the heart of my work, and I kept it like this.
Okay.
Yeah.
Thank you. That's good. Then we can continue with the kidney.
Yeah.
So in chapter 2, you controlled for renal impairment when analyzing your IL-2 receptor beta expression. And then you suggested that is ANCA vasculitis specific.
Yeah.
However, uremia itself in kidney disease is also affecting the immune system. How certain are you that ANCA vasculitis is the cause?
Yeah. So I think what we found is that the beta chain of the IL-2 receptor was sharply diminished on effector T cells, but also on regulatory T cells. And we had a control group in there that also was not uremic, but had diminished renal function. And we found basically that in this control group, the beta chain of the IL-2 receptor was not reduced. So that's why we concluded, okay, it's valid to say that at least uremia is not the most important specific reason why it could be reduced in this case.
Happy with that question. So I have still a little bit of time. So making use of your expertise, because you have a tremendous expertise, I'm very interested in small vessel disease as well, but then in calcification. One of the features of a dialysis patient is that it can enter in what we call calciphylaxis. Can you help me? Because nobody knows exactly what is the cause. 50% of the patients get VKA treatment, so warfarin. Nobody knows what is the underlying cause. Should I look into the immune system? Maybe,
yes. So it's a very severe disease. And I mean, in dialysis patients, you have rapid progressing atherosclerotic disease, and you also have patients that have sharply elevated inflammatory parameters, and both probably contributes to the development of calciphylaxis. And the treatment that we use so far is sodium thiosulfate treatment, but it's not very efficacious. And I also think that we should have a look at the immunological features of this disease. Again, we need trials for that, and we need to see what's going on there. But maybe that's something that could be promising.
It's maybe a nice bridge to collaborate or keep collaborating between Essen and Maastricht.
Yeah.
I'm very happy with your answers. I give the word.
Yeah, thank you.
Thank you. The opposition will be continued by Professor Heringa, Professor of Immunology at the University Medical Center in Groningen. Thank you for coming to Maastricht.
Thank you. Nice to be here. Dear candidate, first of all, of course, I would also like to congratulate you on completion of this impressive thesis. And of course, I would like to extend the congratulations to your famous team of supervisors. So it's clear from the chapters, I mean, this spans translational work of more than a decade, right? And already referred to by the previous opponent. And I think it's really impressive work, mainly focused on T cells, which we will discuss later a little bit. But I would like to start with this nice picture that we have up here, which is the front cover of your thesis. And when I looked at it, I saw the moon, and I immediately thought of Pink Floyd, Dark Side of the Moon. But I think you have a different meaning with that.
Yeah.
Could you explain it briefly?
Yes. So basically, if you look at the kidney involvement in ANCA vasculitis, it is basically necrotizing crescentic glomerulonephritis, and crescentic means half moon, basically, in the pattern that you see. So the proliferative component of the Bowman's capsule is basically a crescent, and that's why I choose a half moon that is seen through a microscopic lens.
That's what.
Yeah, I'm glad that that the idea is so transparent. Yeah.
I also thought of the bright side of the moon because of
Okay.
the contents of your thesis.
Yeah.
But to go into the science, so my first question actually refers to your proposition 5, which I completely agree with. So it reads that new developments in therapies, they rely on true translational research. And I think your thesis is a nice example of that. But in your thesis, you mainly focus on the T cells. So I think 80% of the work is focused on T cells and ANCA vasculitis. So if I would ask you whether you think that ANCA vasculitis is primarily an antibody-driven disease, autoantibody-driven disease, or a T-cell driven disease? So what would you say?
I would say it takes two to tango. So the B cell cannot be fully activated without a T cell, and the T cell is worthless without a B cell. So, of course, it's a disease where the autoantibody is indispensable. That's totally clear. But to form the autoantibody and to get a high affinity antibody, it takes a T cell to dance with the B cell, so to say, to stay in that picture. So,
Yeah. I completely agree, but why then are most therapies focused on the B cells?
well, the B cell is one effector cell that is important, of course, and well, the autoantibody binds to the neutrophil, so the neutrophil doesn't move away from the vessel wall anymore, and then you have the starting point of inflammation, basically. So the antibody alone is, in my opinion, the starting point of inflammation and maybe keeps on driving the inflammation, but at a certain time point, the disease is independent of the antibody formation, and the inflammatory cascade just perpetuates itself.
But
That is also T-cell driven.
But with B-cell depletion, do you only affect the B cells, or do you think you also affect the T-cell compartment?
Well, you also affect the T-cell compartment because the B-cell is also an antigen-presenting cell, and there are some reports in the literature that also suggest that some of the B-cell defining molecules are expressed by T-cells.
Right.
So CD20 can be expressed by T-cells also, to a lower extent, of course. If you look at patients that have undergone rounds of rituximab treatment, they usually also have some sort of decline of T cells.
Right. And I think there's one study, it's an animal study, that also shows that it may affect actually the regulatory T-cell compartment.
Yeah.
So that could be a link to
Yeah.
other work that you have presented here. So another question I have is actually related to the thesis defense that we had this morning.
Mm-hmm.
So of Joop Aandekerk, and he had very nice work on netosis and ANCA vasculitis, and he also got a question on how innate immunity actually links to adaptive immunity in ANCA vasculitis.
Mm-hmm.
I have the same question for you.
Okay, yeah.
How do you think that links, and how would you test it? How would you experimentally approach that?
Yeah. So there have been recent publications that suggest that there is an elevated type 1 interferon signature in the kidney in patients with ANCA vasculitis, but also in the animal model. And basically, there's also a link between type 1 interferons and the T-cell immunity. So in that publication I just mentioned, they assayed basically T cells that have been exposed to interferon beta, and they saw basically that these T cells gained cytotoxic function and were able to mediate tissue destruction. And I think that's one important link between the adaptive and the innate immunity in ANCA vasculitis, also with focus on the kidney, that needs to be unraveled further. However, the effect of type 1 interferons on T cells is really context dependent, because there were also numerous studies in the past that gave conflicting results, like stabilization of Treg function upon exposure of interferon beta versus abrogation of Treg function upon interferon beta exposure. So it's really context dependent.
But if I can interrupt you, I think my question was the netosis itself. How does it stimulate your adaptive immune response?
Yeah.
And does it only happen in the kidney? Could it also happen in lymphoid organs, for instance?
Yes, I think netosis means that you have basically a mixture of DNA, histones, and so on that could act as some sort of that could empower the immune system to recognize antigens. So in that sense, it is also a principle that it's used in vaccination, that you have some sort of adjuvants that basically that basically allow the immune system to establish a stronger immune response. And I think that principle can be transferred also to NETosis. So antigens get trapped in these structures, will be recognized also by APCs. These APCs, upon exposure to NETs, upregulate co-stimulators, and then it's basically antigen presentation to this T cell, and that keeps the immune system going then. So I think it's some sort of adjuvant function.
Exactly, and I think you could test it in your rat model, right?
Yeah, it's a good suggestion.
I think my time is up. I give the word back to the Pro-rector.
Thank you. The opposition will be continued by Professor Levis, Professor of Clinical Immunology at the University Medical Center in Utrecht. Thank you for coming to Maastricht.
Thank you, Mr. Pro-rector. So, dear candidate, first of all, I really like to read your impressive thesis, and I'd like to congratulate you and your entire supervisor team and your family with this thesis and defense today. So, I'd like to start off from chapter 7 on the Th17 expansion in GPA. So, you also briefly showed in your lecture that you find that T helper 70 cells are increased like in many other autoimmune diseases, but there is no significant difference between patients in remission and patients with active disease. Mm-hmm. What are your thoughts about that? Is that what you expected, or can you elaborate on that?
Well, what we expected at first was that we see a decline of Th17 cells in the circulation, because our idea was, and we've shown it in the animal model, that these Th17 cells enter the organs that are inflamed. However, we recruited patients that were in different phases of their active disease. And we know from ANCA vasculitis and especially from kidney disease that there's some sort of switch. So the switch takes place from Th17 immunity to Th1 immunity, and it could be that we had patients that were in a different phase of active disease, and that's why we didn't see anymore a decline in Th17 cells, because they were simply in a more Th1-driven phase of the kidney disease. So that would be one explanation. The other explanation would be that immunosuppressive treatment could have an effect, because recruiting patients with active vasculitis, ANCA vasculitis, they are mostly treated. They have already got steroids. So it could also be that this had an effect on the results that we saw.
So I assume you didn't have sufficient numbers then of untreated patients to...
It was indeed difficult to recruit patients that were untreated in an active phase of disease to make a meaningful comparison, indeed, yeah.
Okay, but then you move towards sort of controlled disease, patients that are in remission, and then you try to correlate expansion of T helper 17 cells to relapse propensity, which you actually could not. Mm-hmm. What are your thoughts about that? So in the second phase of disease?
Well, I think you have these Th17 cells that are circulating in the bloodstream. And we just looked at IL-17A producing T cells, but this is not a homogeneous population as we know nowadays. Back then, we thought it is just one population, but basically you have also within this population cells that had a former Treg function, or maybe cells that still have Treg function. So I think for further conclusions and for further explanations, it would indeed be necessary to have a phenotyping in detail for these Th17 cells, because it's not a homogeneous population. It's very heterogeneous.
Okay, so then unfortunately with your animal model in which you try to target IL-17 selectively, which did not result in a positive response, do you think that you should not target IL-17 at all? Because this is one of your conclusions, right, in your general discussion that you'd like to move towards T-cell specific biomarkers as targets for targeted treatment.
Yeah.
Or do you think a different approach with combined treatments would be better, and that might be the reason why it failed?
Well, if we target IL-17A as an effector cytokine, we are at the end of the inflammatory cascade. So I think we are simply too late. So what we need to do is that we use methods and strategies to target the development of Th17 cells, but we also have to keep in mind that the Th17 cells are not the only cells that are really driving vasculitis. In this case, I already mentioned that there's a switch from Th17 to Th1, and it could be that if you just have an approach with inhibiting Th17 development, that Th1 takes over, and that you indeed see no difference in disease development. So I think the best approach would be to target Th1 and Th17 development at an early stage, indeed.
Okay.
So you could, for example, you could inhibit the developmental factors that are necessary for Th17 or Th1 development. That has also been shown in animal models now, that early interference with Th17 development may ameliorate the development of GN.
Okay, but then maybe a brief question. How would you position then, for instance, agonistic antibodies to stimulate immune regulatory receptors?
Yeah.
Probably not in a context then of targeting Th17 cells at the same time, or how do you envisage that?
Well, agonistic treatment of co-inhibitors means that I fasten the T-cell break so that I'm able to inhibit T-cells. And what we saw in our data is that BTLA, for example, is functional to downregulate Th17 cell responses. The question is always how powerful is that during an active inflammation, because these were ex vivo experiments from patients that were in remission in a non-inflammatory context. And I think that has to be assayed. And maybe it's also an issue for bispecific approaches, so that you really target only specific cells with an agonistic BTLA antibody. That would be one idea. It's a wild idea, so to say, because there are no developments in that field that I'm aware of, at least, but maybe agonistic treatment of co-inhibitors, maybe that's something we should go for.
I think I could go on, but time is running out. I'm happy with your answers. I'll give the word back to the pro-rector.
Thank you. I'm sure you can all continue forever. The opposition will be continued by Dr. Wilde, Associate Professor of Transplantation at this university.
Thank you. First of all, I want to congratulate you with a beautiful thesis. The nice thing of the fact that it's been many years is that you very nicely see progression in the field, and you've beautifully discussed that earlier during this defense. So my compliments also for your supervising team, of course. I'd like to ask one of the para-infant to read Proposition 6.
Tissues are not simply passive recipients of immune protection, but are active participants in their own defense. Polymer Singer.
Thank you. Yeah, great immunologist, way ahead of her time, I think. How do you view this concept in ANCA vasculitis?
Yeah, she's right, of course. So what we see basically is that the kidney is not just a bystander that is just attacked by the immune system, but it's a regulator in its own defense, so to say. So if you look at the kidney during active disease, then we see that, for example, renal tubular cells are able to upregulate molecules that are able to shut down the immune response, like PD-L1. The shame in ANCA vasculitis is that PD-1 is not functional on T cells. So that's why it doesn't work. But in principle, the kidney has its own strategies to limit and dampen immune responses.
And is it also orchestrating the detrimental responses. What is your view on that?
Well, it depends a little bit. So I've talked about the type 1 interferon responses earlier, and what we see is basically that producing type 1 interference is a basic mechanism in kidney immunology. And surprisingly, also renal parenchymal cells, like renal tubular cells, they can produce type 1 interference. So interferon alpha and interferon beta, they do that upon different stimuli. So if you expose the kidney to cold ischemia, you rewarm the kidney, then you see tons of interferon beta. If you look at the kidney during ANCA vasculitis, active glomerulonephritis, you see in specific cases type 1 interferons are produced. And depending on the context, it can be protective.
Mm-hmm. And well, I'm also asking because in chapter 3 you show that there's immature dendritic cells. Mm-hmm. In healthy people they are there as well, but they don't form aggregates. In patients with ANCA vasculitis they do. Mm-hmm. You also suggest that it may be tertiary lymphoid structures that may have a role in suppression or promotion of inflammation. One of the things that they produce is IL-23, which can, of course, induce IL-17 cells. And there's literature showing that blockade of IL-23 in an early stage can, in contrast to your chapter 9, can block progression of disease. How do you envision IL-23 works in your animal model?
Well, exactly. So IL-23 is one of the factors that promotes the development of Th17 cells, and it has been shown in animal models of ANCA-GN that blockade of IL-23 can prevent the onset of GN. Whether this is really confined to the local compartment, whether blockade of the local compartment is the main issue here, or whether the blockade mainly takes place in the lymphoid organs, that's not clearly shown. But I could imagine that indeed blockade of IL-23 also during active disease may have some role in dampening the Th17 response. I mean, the immune system in the kidney that is not primed to defend against autoimmunity. It's primed to limit infection and tissue damage due to other reasons than autoimmunity.
And could it also, to what extent do you think that if such local tissues develop and they promote inflammation, that at a certain moment that becomes more autonomous and sort of the organ starts to orchestrate in a detrimental way its own response and perhaps the transition towards Th1 responses as you described earlier?
Yeah, I think that, of course, that's one thing that could happen indeed. So what we don't see in ANCA vasculitis in the kidney, or what we didn't see in our study, highly organized tertiary lymphoid structures. We didn't see that. But if you look at the lung in this disease, for example, you can see TLOs, so these tertiary lymphoid organs. And in some of these TLOs, it has been shown that outer antibodies are locally produced indeed. And one idea of the rituximab treatment is also that you do not only deplete basically the B cells in the common lymphoid organs, but also targeting these TLOs. However, it has been shown that rituximab is probably not ideal for that, and other treatments like CAR T cells might be more might be more beneficial in that sense. So TLOs exist also in ANCA vasculitis, not to the same degree in the kidney as has been observed in the lung, and can have a detrimental effect indeed. Yeah.
And if you think about the response, there's one study that you also refer to in chapter 9 where they show that ustekinumab can be effective. Mm-hmm. So blockade of IL-12 and IL-23. Mm-hmm. What do you think drives the transition from more Th17 response to a more Th1 response, and can you interfere with that, and how would you do that?
Well, yes, you could interfere with that, but it has already been shown that if you interfere too much with the Th17 cell response, then Th1 takes over, and you have the same degree of damage, basically. So what exactly triggers the switch is not very clear. So it seems to be time dependent. So first phase seems to be Th17, and then the following phase Th1. But if you simply shut down the Th17 response, that's not sufficient, basically. So the exact factors that are triggering the switch from Th17 to Th1 are not known.
Thank you. I give the word back.
Thank you. The opposition will be continued by Dr. Hilhorst, Associate Professor of Nephrology at the Amsterdam University Medical Center. Thank you for coming to Maastricht.
Thank you very much for having me. Professor candidate. Congratulations, and also congratulations to the supervisors. Some of them are quite well known to me. So I was massively excited when, of course, I saw the piece on dendritic cells. So it's innate, and we already briefly talked about it, but they're antigen-presenting cells. You also found macrophages. Are they actually presenting antigen there?
What we see is basically that they express co-stimulatory molecules. And that's why we infer, that's why we assume that they were indeed were presenting antigens, but we did not specifically assay which antigens, and we also did not specifically assay to which extent they could present antigen.
So do you think they're presenting myeloperoxidase or proteinase 3?
Could be, yeah, could be. Yeah, well, I think if you look at lesions in ANCA vasculitis, you see the autoantigen MPO to a certain extent, depends on the disease type, also proteinase 3 over and over, but whether it's really presented by this specific type of dendritic cells, we have not shown.
So where are they coming from?
The dendritic cells?
Yeah.
It's a good question. So we think that they migrate from the lymphoid system or from the bloodstream into the kidney to a certain extent. But of course, you also have resident dendritic cells that were always, so to say, in the kidney, and that they have surveillance function, taking up antigen, presenting antigen, processing antigen, and so on. But we think that the massive amount of dendritic cells that we saw around the glomerulus, so periglomerular, and also in the interstitium, is de novo migrated.
So they come from monocytes?
Maybe, yes, could be. Well, we don't know from which lineage they stem from, but at this time when we were writing this article, and you also mentioned yesterday, the terminology is not very clear on that. There was a very simple definition used for kidney dendritic cells. You had CD209 expressing dendritic cells that were called immature dendritic cells, so DC sign positive dendritic cells, and you also had mature dendritic cells that were, if I remember correctly, defined as dendritic cells that were expressing, I think, CD208 or DC lamp. Yeah.
So imagine you would have a situation of an ANCA vasculitis patient, you would be able to remove all the monocytes. Mm-hmm. What would happen to the disease in the kidney?
Well, I'm not sure, because as I said, there is local regulation going on, and for these type of dendritic cells we found, these were immature dendritic cells, we thought that these dendritic cells may assume a protective role. We have no definitive proof for that, but that's what we assumed. And I think it would be a nice idea to have an animal model where you deplete renal dendritic cells, monocytes, macrophages with clodronate, for example. I mean, that's possible to do, and to see whether you can still induce disease, or whether you have a disease cause that's detrimental or maybe ameliorated. I think that's a very good idea, yeah, to do.
So if they are regulatory, then imagine ANCA vasculitis, which cell arrives first in the tissue? Because then the monocytes are coming in.
Yeah.
So they regulate too late, right? That's what you hypothesized.
Yes, yes. So I think my idea is basically that you have Th17 cells that migrate upon first impact to the kidney, and then everything else that comes follows. So maybe you have neutrophils that enter first, but from the T cell side of the coin, Th17 cells are first. In which position dendritic cells are in this timeline? That's a good question. I think that has not been unraveled so far. Could imagine that they follow the massive wave of T cell inflammation. Yeah.
And there was a recent paper by a French group showing your interferon signature, not only in the kidney but also in the peripheral blood. But it was mostly seen in MPO ANCA, not really in PR3 ANCA.
Yeah.
Do you know why?
Well, that's a good question. So the paper that I was thinking of, it's from the Hamburg group, from Ulf Panzer, and what they've seen basically is that these type 1 interferon signatures of T cells exist in the kidney, and then apparently they also leave the organ and then circulate in the bloodstream. They also observed it in the MPO model, but that's simply due to the fact that there's no proteinase 3 model available. And well, it could be that it's indeed a feature of MPO patients, because we know that PR3 patients are different from MPO patients if you just look at the clinical phenotype. So the MPO patients, they have more fibrosis in the kidney, they have a more smoldering disease course. It's harder to fully stop inflammation in these patients, and it could be that the inflammatory process is just upheld by the type 1 interference. Could be, yeah.
So we should give them anifrolumab together with rituximab.
Well, I think the company would happy to hear that, but I would be a bit reluctant because anifrolumab is antagonizing interferon alpha, and the signatures that we were talking about is mainly interferon beta.
Yeah, exactly.
Yeah.
So one last thing. You mentioned the tertiary lymphoid organs that you think they are there. The problem is that if your antigen-presenting cells are regulatory, how come they can lead to pathogenic ANCA in the tertiary lymphoid organ if they are regulatory?
It's always about balance. I mean, you not only have regulatory or antigen-presenting cells with regulatory function, you also have antigen-presenting cells that drive attackers, the immunity, so to say, and it's always about the balance. And some of the regulatory pathways, as I said, are simply not functional. They are defective in ANCA vasculitis, and that's why you may also see a lot of these regulators in your affected tissues, but that's only due to the fact that they are not as efficacious as they should be because the regulatory pathways are broken.
Great. I have excellent answers. Thank you. I give the word back to the director.
Thank you. The opposition will be continued by Dr. Nieuwhof, Associate Professor of Allergology and Clinical Immunology at this university.
Thank you very much, Pro-Rector. I'd like to congratulate you, candidate, for your very impressive work that you've been working on for so many time, and your co-promoters also, your promoters also, for your very nice work. I also wanted to ask about the Polly Matzinger proposition, as you said, grand lady of immunology, but I do remember she did not have an answer to allergology. So I still have to talk to her whether she can change her mind about that. I wanted to discuss, I think, quite a difficult problem here in your thesis. You've been looking at healthy controls. You have been looking at active disease, not so many people that you were available. Yeah. And you have been looking in remission. Now, I was wondering whether during your work, where you have looked at T cells and B cells and dendritic cells and co-stimulation, whether you could determine mechanisms that you think are responsible for maintaining remission or otherwise provoking relapse. Could you expand a little bit on that?
So factors have been identified that can trigger relapse in ANCA vasculitis, and One of these factors is Staphylococcus aureus, colonization in the nose. We did not control for that factor, indeed, but most of the patients received cotrimoxazole, because at this time it was a standard treatment for these patients, and it was thought that cotrimoxazole was and still is one of the factors that influences the Staphylococcus aureus impact on the disease course. So if you give patients cotrimoxazole, they have if they have a specific type of disease, they have less relapse propensity.
You did look at B-reg cells, and you did look at PD-1, and you saw differences between the numbers of B-reg cells in active disease or in remission. Do you think that they have cues that you can further expand in protecting relapses from relapses?
Yeah. Basically, what we saw is that there was a tendency that patients with active disease had less B-regs than patients in remission. Whether this was causative or not, we didn't know, of course. So what is heavily discussed at this time is cellular therapies, of course, and whether cellular therapies can be applied to patients with autoimmune disease. And with respect to B-regs, there are several problems that need to be addressed. So regulatory B cells have poorly been defined on a phenotypical level. So that's unlike Tregs, so you don't have the specific phenotype of the one B-reg population that could be suppressive, and you have no lineage factor that helps you to identify B-regs a priori before isolation or to check for purity. So these are some technical issues that arise. And the other thing is also that on a technical level, unlike Tregs, you don't have really co-culture assays that you can use to check for suppressive capacity of your regulatory B cells. So these are technical issues on multiple levels that at this time prevent some sort of cellular B-reg therapy that you could think of. Well, patients have not enough, just take out B-regs, expand them, and give them back. In addition, and that's something that is similar to the field of Treg, if you have regulatory cells from patients with an autoimmune disease, you have to assume that there are some sort of defects that you may have not identified in these cells yet, and the question is whether it's really worthwhile to expand autologous regulatory cells of these patients and infuse them back if that really helps. That's not really clear right now.
If I read in your thesis also that interferon gamma T cells differ between remission and relapses. Now, yes. I think you remember, I remembered you saying that it's more interferon beta. Did you look at those ones also?
Interferon beta? Mm-hmm. So we didn't. So in this thesis, we didn't look at interferon beta. So type 1 interferons, that was not the focus of our thesis. The type 2 interferons, also like interferon gamma, we looked on a T cell level, and if we looked at Th1 cells, so interferon gamma producing cells in the circulation, we saw that during active disease they were diminished. And we also thought that's as a simple reason they're going into the tissues, because they normalize once a patient gets back into remission.
Yeah. Okay. I would like to ask you a question on chapter 6. Mm-hmm. This is a sort of an odd chapter out where you studied the effect of chloroquine on SLE, rheumatoid arthritis, and you discussed how that influenced B cells.
Yeah.
I was wondering, should I prescribe now chloroquine for ANCA vasculitis also? Because you did show an effect on B cell, and we give rituximab, also B cell therapy.
Yeah.
ANCA vasculitis.
Yeah. So we looked for chloroquine because it was at this time it was not really known whether there's an effect on B cells at all, and we were interested whether chloroquine may have an undescribed effect on B cell development, plasma cell development, and antibody production. And what we saw basically is that chloroquine indeed, if you reach specific levels, that you can indeed interfere with plasma cell formation and antibody secretion. And we assayed them. We assayed then basically B cells from patients with after renal transplantation, and we saw an effect. But I wouldn't prescribe it now to all ANCA patients. That's the short answer. No.
Benjamin Wilde, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room. Benjamin Wilde, the degree committee here present has assessed the quality of your thesis and the quality of your defense. In view of its positive verdict and taking into account your previous qualifications, the committee has decided to grant you the degree of doctor. Dr. van Paassen, the promoter, is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Dear Dr. Wilde, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Benjamin Wilde, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university. This is a very important moment.
Yeah.
Maybe you can take it. That's a bit better. Oh, you put a seal. I do also take the opportunity to, well, say some words to you. I read in your book a phrase by Livius, better late than never. We can say that together.
Yeah.
I thank the initiators of your project, Professor Quentewaard and Professor Witzke, but I had some time to work together with you to finalize it. It's like a good wine, I would say. Well, you made our day as well. You did really a great job. It was impressive how you spoke to us. It was a lecture, and we learned a lot about regulation. And I would like to congratulate you, but foremost your family, your dear wife, and your 3 beautiful kids. I see one upstairs there. One fell asleep. That's great. Regretfully, you lost your parents over the last years. It was really very sad. But we think of them as well. I think the situation in Germany is a little bit different as the situation in the Netherlands, because you got a first promotion, summa cum laude in Essen, supervised by Professor Witzke, long time ago. You became full professor experimental nephrology in Essen, and you became full professor renal transplant. But now, like Pogacar really wanted to win Milan-San Remo, you finally became a doctor in Maastricht University. So that's a bit like the right order, I would say. I congratulate you there. And as I said, your wife, Sarah. It was actually a sort of fun ride together, because we know each other from the early days when you were a young, brilliant student then already, and working nighttime, weekends, and I was a little part sometime to look together with you for B cells and under the microscope. That was great to be there. You were so clever, so intelligent, but also so modest and so sensitive. I think that's important to note. Oh, sorry. Always working meticulously, counting all these cells, what we learned about. But it was quite a strange time, also for us. And Jan Willem left the department, and then you were gone back to Germany. And there was some interval, and then we got to see each other again at the congresses, etc. And then we thought together, well, you may have to finalize this. And the interesting thing is that after you finish this, I think that we also will find time to get to work together again, because it's important to note that you, apart from all these other more cognitive things, you are also very good to work with. You're reliable. You are also someone that is a giving person. You have a great laboratory. We visited you and we did already some experiments together. So I'm also very intrigued and interested to have a future, and particularly for the younger people in my department, and also for the combination with the work of Peter Hinga, to get in the future more to work together. And having said that, I would like to finish and hope that for today we have a great dinner together and we have a great time together and get time to talk about things in life that are important, like this day together. Thank you very much.
Dear Dr. Wilde, after these very nice words, I also want to add my congratulations, and I can also do that on behalf of everyone in Maastricht University. I think you did an excellent job. Sometimes we have young investigators here presenting their thesis and defending their thesis. Sometimes it's also interesting to hear a little bit information from someone who is mature, has an oversight over many years. What has changed was one of the questions, what has changed, and that's also interesting to hear. We had a discussion about 15 minutes about whether we would grant you the doctorate, and I think the main conclusion was that this is much more than average, higher than average, and I wish to congratulate you with that.
Thank you very much.
Yeah. We are ending this ceremony. After my hammer, I invite everyone in the audience to go to the restaurant to prepare for the reception. We stay a little bit longer here in this area in the stairs to make some pictures, and then we follow you as soon as possible. And hereby I close this academic ceremony.